All right. Morning, everybody. Hope you had a good weekend. I'm looking for Julie on here. I don't see her yet. Any questions before we begin? Let me see. I don't know if you can hear me.
Yeah, we can hear you.
Okay. Now, yeah, I was speaking into the microphone on my computer. I want to try and use my headphones, but thanks. Okay. Any questions before we begin? We'll hand out the exam next Monday after lab on Monday. So about 5:15 Monday, I'll post the exam. There's Julie. All right. Let me make Julie co-host.
I have a question about model complexity, actually.
Yeah.
Does beta 0 count in your parameters for when you're doing that, like 2K? Would you count beta 0? So
yeah.
like for, yeah. Okay, cool.
Oh, good question. The answer is yes. The answer is also, it doesn't really matter. And let's talk about why that is. So you can or you can't. Typically, beta 0 is a parameter, so officially we count it. But let's just talk briefly about why it doesn't matter. Okay. So let me share my screen here. So when we're fitting models and we want to select the parsimonious model based on parsimony, which means we're balancing model fit and model complexity. We have two objectives. We want to maximize model fit and minimize model complexity. We have this function.
Who got yelled at?
AIC equals -2 times the log of the likelihood of our model plus 2K. And here is complexity, number of parameters. And remember, AIC is used to compare models in a model suite or a candidate set of models. Let's not use the word suite. Let's use a model set. And if we think about one of our examples from lab, we had the avian species richness data, or Y, and then we had three covariates, area, temp, and precip. So we had a total combination of, we had three covariates, so that's 2 to the third, that equals 8. And all of our models were beta 0, so these are our values of mu. Beta 0 plus beta 1 times xi1. So here's our model set. First of all, we have mu I equals beta 0, just our intercept only model. We'll call that model 1, model 2, beta 0 plus beta 1 xi2. When we could say xi1 equals area, xi2 equals temp, xi3 equals precip. And just to be totally clear, we could just write area in here, temp in here, and precip in here instead of using x. So we'll say area temp of state I, mu I equals beta 0 plus beta 1 precip, state I. And then we had 4 other models total. And then our final model, model 8, was mu I equals beta 0 plus beta 1 area of state I plus beta 2 times the average temperature of state I plus beta 3 times the average precip of state I. Now, when we look at the number of parameters in these, depending on what type of model we're using, if we're using normal regression model, we have sigma squared, that's in there. But one thing that's common to all of these models is every single one of these 8 has beta 0 in it. So that parameter is always going to be in our calculation of AIC up here. And if we're comparing AIC scores among our models, then whether or not we include beta 0 in our calculation of AIC doesn't really matter, because it's all relative. They're all relative to each other, and if they all have beta 0, they're all going to either get that penalty for K or they're not going to get that penalty for K. So the only thing that matters is if you include counting beta 0 as in your parameters for one model, you do it for all models in your AIC comparison. So actually there's some software out there that do include beta 0 and some that don't. Are in the packages we have been using does include beta 0. And beta 0 is a parameter, so it should be included. But because these are relative to each other, and they all, the penalty is the same for the parameter, no matter what parameter is, if you subtract beta 0 from all of the models, the AIC results are still going to be exactly the same. Okay, any other questions?
Can I just add to that in the context of the lab? Even though it doesn't matter relative to each other, if you're asked to calculate the penalty, still calculate it using beta 0 so that we understand that you know that that is a parameter and that you're counting all of them.
Yeah, thanks, Julie. Yeah, so it is a parameter. Okay, a couple announcements. The exam, I'll post that around 5:15 on Monday next week. You'll have till Friday to do that. Lab 5, I think it is posted. It's been posted since last Wednesday. The partners for today are posted on that. So that will be due the day of the exam.
For the test, is it in one go, or can you exit out of the page?
You can exit out. Yeah, I'm just going to post the exam, and you're going to work on it independently and answer the questions. Now, I posted the solutions to Lab 3 in there, and the type of solution and the type of detail that we're looking for in an exam is going to be similar to what the solutions are for Lab 3, and then we'll post them to Lab 4 as well. So if you're looking for examples on, what material or what content that we expect, look at some of the past solutions for, especially from Lab 3 on. Okay, another comment on working in groups. Working in groups and collaborating in general is a really important skill to have. And that's why we do it in this class, because these types of projects you almost always work on with other people in the real world. Rarely do you write a manuscript. If you look at all the manuscripts that are published on wildlife ecology now, there's almost 0 manuscripts that have a single author. There are some, but it's very rare. So we're always working with other people to do this. If you're assigned a partner and your partner reaches out to you and you don't respond, expect to get a 0 on the lab. if you don't turn it in. Okay. Exam will be due Friday. I think I'll say before midnight Friday on after that week. So you'll have part of Monday, Tuesday, Wednesday, Thursday, and Friday to do it. On October 9th is when it'll be due. Lab this week, Monday, let's see. Monday, today's lab, we'll go over working on lab 5. But after that, we won't have additional lab material until after the exam. So lab 6 will be after the exam. We will use
Sorry, are you still meeting on Wednesday and Monday then too?
Yep. We'll still meet in lab on Wednesday. We'll go over material. We'll go over interpretation. We'll review all of the material we've talked about. There won't be any new material, but we'll cover expectations and guidelines for addressing questions. So that's why the lab will be posted after lab on Monday, the 5th, because on the 5th, we'll do the exact same thing that we do this Wednesday. So both labs will have that opportunity to review the material. All right, any other questions on those general announcements?
I do have a question. You said it was starting at 5:15 Monday and it goes till what time on Friday?
Let's say midnight Friday.
Okay.
If they're postmarked by, if the emails are marked by midnight on Friday, we'll accept them. And then we'll review them after that. The Wildlife Society annual meeting's happening this week. Things are particularly busy. This is the annual conference that most wildlife biologists are members of. It's all virtual. It was scheduled to be in Louisville, but it's all virtual and online. I see Julie and I both have presentations where that maybe we could post those on the class website too. It's just this, if you're interested in seeing kind of where the types of things that people present at those meetings.
Can I ask you a question?
Yeah, go ahead. Yeah, go ahead, Nicholas.
Oh, it's okay. I think I figured it out. Never mind.
Oh,
thank you.
Okay, sure. All right. So let me see if I can get this to flip around here. Nope, that doesn't help. Keeps its orientation. All right, so here's a schematic of what we've talked about so far. Just a high-level overview of what we've done. And the main question that we try to address in this class is, how do we use data to answer questions about wildlife conservation and management? So we all kind of know data are important, and that's how we can learn about the real world. But the question is, when we have these data, what exactly do we do? What are the methods and tools that people are using in wildlife science to answer these questions that we have? So in previous classes, we've talked about formulating hypotheses and testing hypotheses, and how to look at study design and collect some data to answer questions. And then once we have that data, how do we go beyond something that we would typically do or that we might do in Excel to a more real life analysis? So we talk about several tools that we've used, or that are most common in wildlife analyses. And most of these tools are some form of generalized linear model. So that's what we've been talking about the first 6 weeks of this class, the first nine or so lectures. And how do we write out those models? How do we interpret those models? What does it mean when what our parameter estimates are returned to us? How do we take data and fit those, our models to those data in R, and then know what to do with our output? So generalized linear models are the most widely used types of models that in all of science, really. And these models, what we use them for is to describe a relationship between a response variable y and then predictor variables x. We've talked about 3 types of generalized linear models. Linear regression, where our y data can be anything between negative infinity and infinity. Bernoulli regression, when we have zeros and ones. And Poisson regression, where we have count data. And of the 2 most common types of data that we see in wildlife, it's typically zeros and ones and counts. And there are a huge number of data types that fall into these categories, and some basic form of either Bernoulli regression or Poisson regression. Now, we've talked about the basics, the principles of these models quite a bit, and how we can interpret our parameter estimates beta in terms of real life scenarios. So now we want to look at how we actually use these tools to say something about population size. And we've talked about this already throughout learning about these tools and how we can interpret these models for examples. So we looked at several examples and how we apply these models to those types of examples. There were 3, a lot of the differences between these type models can be described in what type of link function we have. With linear regression, we have no link function, meaning we have our expected value mu I equals just beta 0 plus beta 1x. We don't have to do anything to beta 0 plus beta 1x to get our expected value. With our Bernoulli regression and our 1,0 data, we have to make a transformation of our beta 0 plus beta 1x. And that transformation is the inverse logit function, and it's described by this equation here. So given parameters beta 0 and beta 1 and covariates x, can you describe what the probability of success mu is? And then Poisson regression is actually a little more simple than the logit function. We have a log link function. And to get from our parameter estimates beta 0 and beta 1 to our expected value of our counts, we just take the exponent of this whole combination. So R will give us beta 0 and beta 1, and we have to know what to do with that to give us the expected value, mu I. All right, so let's use these tools to say something about population size. And we're going to talk about methods for estimating population size in today's lecture. Okay. So this is the beginning of a new chapter. This is lecture 10. And it deals with questions related to how can we estimate how many animals are in a certain area during a certain time. And this is often referred to as N, big N. And this is a huge question in wildlife conservation and management. One of the first questions when thinking about some population is how many are there over a certain range. So what is N? So here are some questions that we can, initial questions that we might ask when we're thinking about population assessment. First, how many are there? What is that N? Next, what are the dynamics of that population? So how is the population changing from year to year and from location to location? And although this is a simple question, this is a huge area of wildlife science. And developing methods and tools for estimating how populations change in space and time is one of the primary objectives that my research lab consists of answering. And there's a lot of complexities and idiosyncrasies with every population, and developing methods to address those complexities and idiosyncrasies can sometimes be complex. Why is the population changing? It's another huge question, because if we know why it's changing, we can, if it's changing for the worse, we can try and do something about that. And this brings us back to our basic model of population dynamics, where we have our BIDE model, where our population at time t equals the population at time t minus 1, plus however many new recruits are added to the population, plus how many animals come from somewhere else, subtracting off how many animals died that year and how many animals left to go somewhere else. So with this simple model, it covers a huge area, not only of population science, but also theoretical ecology, movement ecology, and lots of other questions. So once we know something about why the population is changing, we can start to ask what might happen if we conduct a management action. So if populations are decreasing, like in the case of Henslow sparrows, and we need to implement management to try and improve the habitat, if we improve the habitat, are we going to increase the birth rate? And so we can think about things like prescribed fire. Predator control was a famous one in the early 20th century, and removing wolves to try and improve game populations. And then habitat improvement. So we're going to cover a few methods of how we actually estimate abundance. And all of them are extensions of what we've already talked about. Some of them are the exact same methods we've already talked about already. And some of them we're going to add in a little complexity to deal with things like detection probability. So we're going to first get to detection probability, a huge area in wildlife science, when we talk about distance sampling. The good news is nearly all of these methods, like I already said, fit within the framework we've already talked about. So this is why it's so important to have a solid understanding of the material we've covered already. And so that includes normal regression, binomial regression, Bernoulli regression, and estimating covariates. When we get to distance sampling, we're going to talk about detection probability. When we get to mark recapture, we're going to talk about detection probability. All right, the first one on here is complete census. And complete census are rarely feasible in wildlife populations, rarely if ever. Complete census basically means we try and count all the animals out there. This year is a census year for the US. The US is trying to count every individual in the US and try and get some parameters, to also get some covariates about that population. The US Census Bureau is often criticized by statisticians because we seldom need census. Oftentimes a good sample will do. All right, so here's some general notation that we're going to use throughout the class. We're going to let n equal our population size. That's going to relate to abundance. So abundance and population size I'll use interchangeably. They mean the same thing in this class. A is the area of our study region. For our example here, our area is 5,000 square units. And then D is going to be the density, and density equals the population size divided by the area. So a census, we're counting everything. We count every single animal in this area. Animals are represented by dots. You could see immediately why this would be problematic with wildlife populations. Wildlife populations move around a lot, and we can't cover an area fast enough to count everything. In some cases, we could get something like a photograph, but usually the population's spread out over a wide enough area, and there's issues with resolution that photographs cannot help us count everything over the area of interest. So with sea otters in Glacier Bay is about 100 kilometers in length. You might be able to take a remote image of the entire Glacier Bay from satellites. However, counting all of those dots within those images would be challenging. Okay. So censuses are rarely practical in practice, but this is, if we could estimate population sizes perfectly, this is how we would do it. The next thing is plot sampling. So I introduced census. We won't talk about it the rest of the course. What we're going to do the rest of talking about estimating abundance are the other methods. So plot sampling. Plot sampling assumes that we can see every animal within our plots. So in this sample, we have transects set up within our same population we were looking at before. And these transects are in the green area. A person would walk along these transects and they would count the number of animals they saw within the transects. Here we see the red dots represent the animals that were caught. So if we know something about how many transects we walk, the length of the transects, the width of the transects, and the area covered over the region, we can say something about little n, which is the number of animals that we're seeing, the number of animals that were counted, and use all of this information here to try to estimate how many animals are actually in the population. This type of sampling and estimating of abundance is called design-based estimation. That means we design a study. Specifically, we design sample locations. Within those sample locations, we can count everything perfectly. And if we know the area we've covered and we know the population we're interested in, we can do some calculations to estimate how many animals are there. Okay, so how would we estimate abundance from this? I'm going to go to group here. Okay. Stop share. Actually, I'll leave the share up. All right. How do we estimate abundance given these data? Who has an idea?
Do we take the amount of animals known in the entire plot and not just the strips?
So the question is, how many animals are in the entire plot? Yeah, let me lay out this question a little better. So question, how many dots are there? How many elk are in a management unit in Reno? Or how many bears live in the Tahoe region? That's the kind of question. So we have some boundary defined, some box. We know there's some out there. The question is, how many are there? That's unknown. So big N is unknown. What we can do is develop transects to go through those areas and count how many we see along those transects. And then Brianna, to get back to your comment, what we're trying to get at is how many total are there? So what is big N? What we know are the red dots and the area of the blue lines, blue or green lines. What we don't know are the black dots. How can we get at the black dots?
Do we add some of those up and then put it all over A, little a?
Yeah, that's a good start. Here, let me switch my screen here. So let's say add them up, you mean count the red dots, right?
Yeah, but also like the L and the K and the W.
Yeah, okay. So let's see, let me switch my screen here. And see if I can find. There we go. All right, now I can put these next to each other. All right. Red dots, we'll say that is little n. We counted 36 of those. All right, now, Leela, what were you saying about adding up L and A? I think A.
So I thought it would all, you would add it up and then it would all go over little a because that's the area that we covered, right?
Okay, okay. So
But
let's
now
see.
now I see that the A has the W and L, and that, so now I'm a little confused.
Okay, so little a is the width times the length of these transects times 2, because W, the width, is just half of the width. So 2 times W equals the width, and then L equals the length. So if you want the area of a rectangle, we do length times width. And that gives us 2 times W times L, which equals 500. So each one of these rectangles is 500 square units. And if we've done 5 of them, 1, 2, 3, 4, 5, so we have 2,500, is that right? 500 times 5 equals 2,500 units squared. So that's how much of the area we've actually covered. And in that area we found 36 animals. All right. One way to think about this is the number of animals we've seen is little n, and big N is the total number of animals. So we've seen only a fraction of the animals, right? We don't know what big N is. This is what we're trying to figure out. We also know, we do know what the area is. Let's see, in the previous slide, we said the area was, let's see, I'm getting some feedback. Okay, are you hearing me twice? Is there a huge echo? No, okay, just me.
You're good.
All right, I'll try
Yeah,
and deal with it. It's like I say something and it echoes back in. All right, we know the big A equals 5,000. So we've covered roughly half of our area we've surveyed. Okay. So the ratio, if we assume the ratio of the animals we've seen over the total number of animals there are equals the proportion of area we've surveyed divided by the total area, then we can do some rearranging of this equation to try and get n by itself. So if we do some algebra, we can multiply We want to get this by itself. So we can multiply both sides by n. And that gives us n equals big N times little a over big A. And now we can just divide both sides by this number. And that's going to give us n over little a over big A equals n. Another way of writing this is little n times big A over little a equals n. Okay, so that's how, if we use a Planck sample, we know the area that we've surveyed and the total area of interest, and how many we actually saw, we can estimate abundance. Now there's a number of assumptions with this model. So what does this assume? Let's write these down. All right, any ideas?
I had
We're assuming. Never mind.
Brianna, we'll come back to you. What was your question?
so I was just looking at the example on the left side, and I was just a little confused when it says like I saw 36 animals and I covered 500 out of 5,000. Wouldn't it be the 2,500 because we saw 36 total?
Yeah, there should be a 2 here.
Okay. Okay.
Let me,
makes sense. Thank you.
let me go back. Where did I get 500? Oh, no, okay. This 5, I think I multiplied it by 5 twice.
Okay.
So I said 250, and then I multiplied by 5 for each strip. But really, I had already done that. So just to clarify, let me clarify this example. Each one of these strips is 50 meters, we'll say, 50 units. There's 5 of them, so the total length is 250. The total width is 2. 1 represents half of the width here. So then 500 is the total area we covered. When I said 2,500, I accidentally multiplied this by 5 twice. But it's
Okay.
just 500. Yeah, thanks for that, pointing that out. Okay. So I saw 36 animals. I covered 1/10 of the study area. And then to estimate how many there are, we make this calculation that we just went through. And it says there's about 360 animals. All right. So let's go back to Brianna. What does this assume?
Well, it's certainly assuming that there's an even spread, or at least that you're not going to see a higher density than you covered, or like no animals in a one maybe bad habitat area.
Yeah, exactly. So the way we would say that in terms of model assumptions is every animal has an equal probability of falling within our survey area. We'll say being sampled. All right, what else does it assume?
Are we assuming that we see every individual?
Exactly, yeah. If it's on our transect, we assume we see it. We're going to talk about detection probability. And what this assumption is in relation to detection probability is that our probability of detection equals 1. This is rarely the case in wildlife. This works well for studying humans sometimes, but it's a big issue in wildlife science, that whether we see every animal that's within our transect. Animals don't like to be seen. And actually people, if you send them a letter, a survey letter, rarely do everybody respond. So if you send out a questionnaire to everybody, a lot of times people won't respond. If you're standing outside a polling center about a month from now and people are walking out if they voted, a lot of times people won't want to talk to you and answer your questions. Same idea with wildlife and human polls. And we spend a lot of time trying to think about how we address that. All right. That covers the assumptions that we want to talk about. I also mentioned that we're assuming this relationship. Let's see. I drew it out. We're assuming this relationship here. And this isn't always going to be equal. And that's why we're going to have estimates, and we want to account for the uncertainty in those estimates. All right, so that talks about, that's how you do plot sampling. And similar to census in wildlife, rarely can we do plot sampling, mainly because of this issue of 1, detectability, and 2, movement. Animals move around from one site to the next. All right, so this is our first example where we're going to introduce the idea of detection probability. It's called distance sampling. This was developed by 2 groups in, one in the US at Colorado State University, and one in St. Andrews, Scotland. They talked about developing methods for estimating detection probability when you're surveying from a point. And the general idea behind, I can only I can only say a few words, and then I have to pause because I have to listen to myself in this ear. It reminds me of Lou Gehrig, and today I consider myself. Anyway, distance sampling is an extension of plot sampling where not all animals in the covered region are detected. Here, W equals 2. The width of the transect is wider than our previous example. We can make our transects wider when we use distance sampling compared to plot sampling, because we're going to start off with the assumption that we're not going to observe all individuals in the plot. If we were doing plot sampling, we would have to make our the width of our plots only wide enough so we guaranteed that we saw all animals in the plot. Now we can make it wider because we're not going to assume we see all of them. If we look at these plots, there's both red dots and black dots within the blue-green area. The red dots are the animals we see. The black dots are the animals we don't see. So you can think of these in terms of someone walking along a road, counting Henzo sparrows in a field, someone flying in a helicopter, observing elk and trying to count the number of elk, doing frog surveys if you're walking along a road, listening to calls from ponds where frogs are calling. So we can use these types of surveys for a lot of different methods, a lot of different species and populations. All right, so here's an example. The strip width here is 2 on each side of the line. The area of the region covered in this specific example is 1,000. We can calculate that area by knowing how wide the strips are, how long the strips are, and how many transects we do. And then we have a number of animals counted. But we know now, not only are we not counting animals that are in the transect, but we're also not counting animals within the transect. And we want to estimate how many animals there are total, all of the red and black dots here. P is going to be the proportion of animals detected within the covered region. It's how many of the dots are red in the green blue area versus how many red and black dots there are in the green blue area. So for now, imagine we know that detection probability equals 0.7. This is related to what we've already discussed in Bernoulli models, where probability of success equals some value between 0 and 1. Now let's assume that probability of success equals 0.7. Let's go back to our basic Bernoulli model, where we have our data, yi, our whether or not we saw an animal I, and we have some probability of success P. If we know what our probability of success is, 0.7, we can estimate what our beta is. And using the inverse log function, we can calculate that as 0.84 using all of the equations that we've already talked about. In terms of interpretation, the probability of successfully observing an animal in our transect is 0.7. Now in practice, we're not going to know what this probability of success is. We're going to have to estimate it using our data. Okay, so this is mainly a review of how we go back and forth between our probability of success and then what our parameter values are. So when we run a model in R and it gives us an estimate for the intercept, and that intercept equals 0, after we go through this function here, this inverse logit function, it's going to tell us that the probability of success is 0.5. Now, what we're going to start off with in our next class is going from our Bernoulli model, which is one coin flip, to a binomial model where we have more than one coin flip and estimating the probability of observing the results that we actually got. And I'll stop there on this slide. Any questions?
I have a very quick lab question.
Yeah, go ahead.
So I was talking with Julie about this, but for question, I'm never going to remember what they are. For question 1, I forgot where it was. Do you want the general model statement or do you want the R code hypotheses?
Lab 4 or 5?
4, sorry.
Okay. And question one, did you say question one, right?
Yes.
Develop a set of 10 appropriate candidate models reflecting hypotheses about the relationship between habitat selection by spotted owls and the covariate information that is available. Describe your hypotheses for each candidate model. Okay, this is an important question because this is where we start to incorporate our science into this class here. So I'm going to pull up the solutions for lab 3 to show an example of how you should address this question. All right. Where'd it go? There it is. So this was lab 3, question 3, part I. It says develop a set of at least 10 candidate models for the spotted owl home range data. So very similar idea to the question in lab 4. And here's what those solutions look like. We have 10 models. Each one is described by a bullet point here. Our first model is the null intercept only model. That doesn't need a lot of description. The second way we would say is, so let's go to our model first to see what model we're describing. Our first model is the intercept only model. Our second model describes home range size to the proportion of mature habitat so this is old growth mature forest within a spotted owl home range. So to introduce that model, we say something along the lines of this. We assume that small home ranges reflected better habitat conditions or configurations than did large home ranges, because the higher the quality of the habitat, the shorter the distance an owl would need to travel to meet its requirements. Therefore, we hypothesize that spotted owls having more mature forest within their home range would have smaller home range sizes, because mature forest has been presumed to be their primary habitat. And there's sources to back this up, what people have found in previous research. I would say for this class, and this lab particularly, you don't necessarily need to find literature to support your hypotheses, but it's a good place to start and to think about what hypotheses you want to develop. So this is an example of what like a 100% solution would be on a lab assignment or an exam. We're not requiring this detail for lab 3. But moving forward, we want to start to work up to these type of hypotheses. Does that answer your question, Brianna?
My question was actually more of when writing the candidate models, did you want them written in R code or written them as a model fit statement?
So the way I would do that is write out, let's see. What type of model was this?
Bernoulli.
Bernoulli model, okay, good. So here's how I would do that. I would write out and so on. So you have your general model statement up here, and each one of your different models represents different covariates and combinations for mu. Now let me look at that question again, lab 4, question 1. Okay. Yeah, so the first part of your question was related to writing out this part here. But the part I talked about before is describe your hypotheses for each candidate model. Any other questions?
Yeah, Perry, I have a quick question about lab 4, question 7. I just was thinking that for answering that, you fill in the AIC value and then you plug in because we know 2K and we know AIC and we're trying to solve for -2 log. So do we just plug in what we know and then solve for it?
Exactly. That's algebra.
Okay, that's what I was thinking. So yeah, we just have our AIC value. Okay, cool.
Yeah, you have two knowns and one unknown. How do you solve for the unknown?
Right. Okay. Yeah, and that's what we did. We just wanted to double check with that.
Okay.
I had a quick question about lab 5.
Yeah, let me pull that up. Okay, go ahead, Nikki.
So for the very last question where it talks about estimating if it was 20% of the total area, if our model, our top model, our only covariate was time categorical, and so I'm assuming the 20% total area doesn't apply to our model since none of our covariates involve any of the other data besides time.
So this is similar to what we talked about today, that you're going to get an estimate of how many sea otters there are, but we only covered a certain range within that area. So let me draw out a picture. First, I'll write out your model. So tell me if this is what you got. You had our data, our counts of sea otters, came from a Poisson distribution with expected value lambda, and then the log lambda equals beta 0 plus beta 1. Did you say time was continuous or categorical?
Categorical.
Okay. So you had time 2 plus a lot of time periods. There's like 20. Do you know how many there were?
There were 22 levels is what R said.
So beta 21 times time. Now this, what you're be able to do is calculate what y is for each site for each time period. I mean, not y, I'm sorry, lambda. So lambda for site I is going to equal some beta. So for the very first site, it's going to be beta 0. Now you have 1 to n data points, right?
Mhm.
So you can calculate lambda 1, lambda 2, lambda 3, all the way to lambda n. That's the expected value for each site. So to get the expected value for the entire Glacier Bay, you first have to figure out what the expected value of your sampled area was. So you have the sum of lambda from I equals 1 to n, and this is just basically saying lambda 1 plus lambda 2, lambda n, right? And that'll give you how many sea otters you expect to find at each area you sample. So if we break this up, and a helicopter flies over here and there and collects data at each one of these cells. So here's 1, 2, 3, 4, 5, and so on. You get counts, or you get data at each one of these cells. Then you're going to get a lambda 1, lambda 2, lambda 3, and so on. So you add all these up, and that covers the total survey area. Now, if you want the estimate for the entire bay, we're going to have to do exactly what we just did in class. So we have little a over big A equals this value is going to be your expected value of n. So you could say the sum of lambda I from I equals 1 to n divided by big N. And this is what you want. We know the problem told us that this equals 20%. Any questions about that?
And then I'm kind of confused in R when we run the factor function to do the categorical. So when we do the summary of that model, because when I wrote it in R, I just did factor of time, the time category in the data. And then that gave me, I input that in our fitted model and I fitted it and it gave me a summary. So I don't have explicitly written out like levels 1 through 22. I assumed R does that on its own. So the summary of that model is representing lambdas for 1 through 22 already.
It gives you betas, and then you have to take those betas and convert them into lambdas.
Okay.
And we talked about this in lab on Wednesday, right?
Yeah.
So right after we talked about it, categorical variables, I got the exact same question. And I thought a lot of people are going to have this question. So the next time I recorded it, and then I posted that recording online. So if you want to talk more about going from our summary on categorical variables to calculating lambda, there's a video on that. Have you seen that? Yeah.
No, that was posted after our lab on Wednesday.
I posted right after our lab on Wednesday.
Oh, okay. Is it just called like Lab 5? Or
There's, it's called something like categorical versus continuous interpretation, but it's under lab 5. Yeah.
okay. And then my last question was just for the answers for lab 2. Are those posted?
Yeah, they should be. Let me check. I'm fairly certain they are, but I'll double check. Oh, they're posted, but they're not published. Okay. Thank you for pointing that out. I had to click a little green arrow next to them.
Awesome. Okay, well, thank you.
Yeah.
I had a question on number 8 on lab 4.
Okay.
I'm kind of lost on how to do it.
Oh, okay. Covariant top model. Let's talk about your top model.
Okay, so my top model, it's number 6.
And this was, which data set was this?
The
Spotted
owl
owls. Okay. And model 6 could be different for every group.
Yeah.
What was your model 6?
Here, I could share my screen if you like. Oh, wait, I can't.
Oh, okay. Let me make you a co-host. Okay.
Okay, so this is what I got.
Model 6, you include distance to water. Okay.
Yes.
So beta 0 equals negative 1.392.
Yes.
Beta 1 equals,
That's the distance water, right?
yeah, 0.00028, let's say 289.
So that's how you read it, right? Because it says 2.887.
Times 10 to the negative 4th.
Yeah,
So you have to move that decimal place 4 spots over. So let me share my screen.
Well, that's correct, because I put it up on there.
Yep, that's right.
Okay.
Okay. So here's your model right here. And the question is, choose specific covariate values for the covariates in your top model. This will change depending on which models you considered. For you, it's distance to water, and calculate the probability of selecting that habitat. So the probability of selecting a specific habitat is the same thing as the probability of success. And where do we measure the probability of success? It's right here. It's this mu value. So we have to figure out what mu is.
Okay.
And it says, choose specific values of your covariant. So your covariant is this distance to water. So what we have to do, let's say we're interested in the probability of owl using a site that is 100 meters away from water. So we go through and we say, the logit of mu I equals beta 0, which is negative 1.392 plus 0.000289 times water, and we're going to put in 100 there.
Oh, so we just put a random value.
A value that you choose. So whatever one you choose. Yeah.
Okay.
You could choose 100 or 50 or 0 or whatever.
Okay.
And then figure out what mu is. Do you want to go through how to do that?
Yes, please.
Yeah, okay. So to get mu by itself, we have to get rid of this logit function, which means we have to take the inverse logit of it. So we do the logit inverse of the logit of mu equals the logit. We have to do the same operation to both sides of the equal sign. So we're taking the logit inverse of both sides of this equation here.
Okay.
So that is the logit inverse of negative 1.392 plus 0.000289 times 100. Okay, this and this cancel out. So now we have mu I equals the logit inverse of negative 1. Do you want to just, do you have R open?
I do.
Can you calculate what this value is in here?
Okay, yeah. Am I supposed to? How am I supposed to write that in R? Because I just put it on here and just didn't want to.
Yeah, let me share my screen.
Sorry.
We can go through that too. So can you see this down here?
Yeah.
Okay, so we type in our console, negative 1.392 plus 0.000289 times 100.
Oh, oops. I put the parentheses instead of the.
Oh, right. Yeah, it doesn't like parentheses for a
Okay.
multiplication sign. So that gives us negative 1.3631.
Mm-hmm. I got that.
And now we just have to figure out what this logit inverse function is. And we've talked about that. That equals the exponent of this
Oh, wait, I can't see it.
divided. Oh, sorry. Let me change back to. All right. So we have the inverse logit of this. This function actually means the exponent of something, whatever's in this parenthesis area, the exponent of this divided by 1 plus the exponent of that. So we can write that out. The exponent of negative 1.3631 divided by 1 plus the exponent of negative 1.3631. And that's going to give you your solution.
Okay.
That'll give you mu, and that's what we're trying to get.
What do you have in front of that exponent? It says exponent. Oh, there, 1. It's 1. Okay.
Right here, yeah. 1 plus.
Negative X. Okay. And then I could do that in R, right? And it'll calculate.
Yeah, let's go through that.
Calculate that for me. Okay. Let's see.
So we say the exponent of negative 1.3631 divided by, now we're going to make sure we want to put parentheses here, just so we have the order of operations right.
Okay.
because we need this 1 plus negative 1.3631.
Oh, we don't put 1 plus the exponent?
Oh, yep. Nice. I was wondering why I got a negative number. And this should always be between 0 and 1. There we go. So that gives us our mu value, what tells us the probability that an owl will use that site 100 meters from water.
Okay. I don't know why I'm not getting that. It just has a plus sign.
Oh, you need another parenthesis. So you have one, you miss one of your parentheses.
Oh.
When you get that plus symbol, it means it's still waiting for you to finish the command.
It says there's an error.
Do you want to share your screen?
Yeah. Okay, so I did this, right? Is that not right?
I don't see your screen yet.
Oh.
That looks right.
That's right.
Yep.
Oh, there you go. I don't
There
know
you
why
go.
I tried it here and it didn't want to work. I'll
It
probably
looks
put that.
like up above. Well, it's because you still have that plus symbol.
Oh, that's why. Okay.
It has to have that caret or greater than sign for it to work.
Okay. Okay, thank you.
You're welcome.
That helps so much. I was like stuck on that for quite a while because I didn't understand what it was trying to tell me.
Oh, yeah. That's a common one. Probably see something like that on exam.
Okay. Thank you.
You're welcome.
And that's all the questions I have.
Okay.
Thank you.
Yep. Have a good day.
Yeah.